Go.
Okay. All right, so now we're continuing with selective and differential media. Now that you've taken a minute to figure out which plates are selective and which are differential, now I'm going to talk about how to actually read the test results of the plate, because these are the kind of tests you would be doing if you were a lab tech in the hospital. So the EMB plate, that's the one that's purple. It selects for gram-negative rods. That means only gram-negative rods can grow. It's testing for lactose fermenters. Wants to know if you're a tummy bug. So it's a weird plate. It has three possible positives. Either they look like purple fried eggs, or if you're thinking, it doesn't look really purple, I'm not sure, turn the plate over, look from behind. If it's dark from behind, that's also positive. Or it could be metallic green, depending on the bacteria. So those are your three choices. It doesn't have to look like all three, it just has to be a purple fried egg, dark from behind, or metallic green. And here's a couple of plates to show you. There's one. This would actually be dark from behind. And here's a metallic green. So those two are positives for an EMB plate. Okay. Next is the mannitol salt plate. For the mannitol salt plate, gram-positive coccus can grow on it, but it's actually testing for Staph aureus. So Staph aureus, if it grows on the plate, it actually turns it yellow. So yellow agar is a positive result, but it also means you have Staph aureus. So I don't know if you remember, but the plate was red last week. Now it's red and yellow. So where the bacteria is growing is now yellow. Okay. Next, the MAC plate. So the MAC plate and the EMB plate are similar. It's just a different dye. So on a MAC plate, your bacteria is going to be pink instead of purple. But otherwise, you should have matching results. Your EMB plate and your MAC plate should have the same things growing on them. And here's the MAC plates. So here's C. frundii, here's E. coli. So it's different kinds of pink, so you can tell the difference between them. They're two different kinds of pink. Okay, and then unfortunately, I don't have a blood agar plate. I have those at home, but for the blood agar, there's no positive or negative. It's based on what they call hemolytic groups. So it's based on how much blood they eat. So there's three categories. An alpha, which I call the blood snackers. They can eat the blood, but they can't break it down completely. So it gives them a weird green color. Then there's the betas. For the beta, you take your plate and you hold it up to the light because you're looking for a clear zone. So the alphas and the betas look alike on the plate, but when you hold it up to the light, that will tell you the difference. The third group are called gamma. They barely grow. They're not blood eaters at all. So when you take a look at the four plates, you'll notice one of the four that are growing, one barely grows. That's the S. faecalis. So they don't like blood. Okay.